Okay, so I just want to run through respiration. Let me see. So you've talked about respiration. You've talked about starting off with glucose most of the time. So this is going to be a quick recap. Whoops. And we're just going to summarize glucose to pyruvate. And let me see. Oh, whoops. Glucose to pyruvate. That is glycolysis, obviously. And can anybody tell me, Jessica, can you tell me how many ATP are made? Oh, hold on. How many ATP are made?
2.
Yep. So I suppose we'll be a little bit, we'll say 2 ATP are used, 4 are made, and so that gives us a net profit of 2. Anything else made during glycolysis, Sarah? What do you think? You might have to unmute yourself.
2 NADH.
Yep. So we'll put that to NADH there. So that is glycolysis. You end up with pyruvate. How many carbons in glucose? Ellie, how many carbons does glucose have?
6.
Good. And pyruvate? Ellie, yes. So keep on with you there.
3.
Great. So you know that obviously the glucose splits in 2, so we've actually got 2 of those. Right, so if we move that up out of the way, what's the next stage, Grace?
The link reaction.
Okay, so glycolysis, just see what color I'd written in. So that's the link reaction. Jessica, what do we get? Pyruvate converted into
Acetyl coenzyme A.
Yep. And how many carbons?
2.
Yep. So we have 2 carbons. Who else have we got? Chantel, what is sort of produced as a byproduct there? Or what's produced during that link reaction?
Carbon dioxide.
Yep. So we've got carbon dioxide. And is there anything else that comes off? I'll go back to Chantel there. Anybody?
NADH.
Yep. So we've got more NADH. What comes after acetyl coenzyme A in the link reaction? Who have we not? Let me see. Bethany, what comes after acetyl coenzyme A?
I don't know.
Anybody want to help her? Ellen, Farquhar, your name has appeared up on my screen. Anybody?
Is it the Krebs cycle, Miss?
It is. Who was that?
That was Hannah.
Very good. So that's the Krebs cycle. And we're not going to go through all the different stages, but what do we end up with for every Krebs cycle? Well, in the matrix of the mitochondrion, there will be a 4-carbon acid which joins to acetyl coenzyme A, which has got 2 carbons, so it makes a 6-carbon compound. That's then converted into a 5-carbon compound, which is then recycled into the 4-carbon compound, which is ready to pick up the next acetyl coenzyme molecule. So obviously, when we're going from 6 to 5, we're losing carbon dioxide, just like when we went from 3 pyruvate up here to 2 down there, we lost a molecule of carbon dioxide. Whoops. So we're going to get a molecule of carbon dioxide here, and we're also going to get another molecule. Whoops. You think I've been doing this for months? You think I would know what I'm doing? Get another CO2 coming out there. Dear me. Okay. As well as carbon dioxide, what else do we get being sort of picked up or produced? Ellen Black, what do you think? Any of the Ellens? Ellen Hamilton?
FAD.
Yep. So FAD it's really like a hydrogen carrier. It will pick up hydrogens. So FAD, oh dear, I'm going to cry. FAD picks up 2 hydrogens and becomes FADH2. What do we call it when something picks up hydrogens? We say that it is what?
Reduced.
Excellent. Thank you, Grace. So FAD is reduced. What else is reduced?
NAD.
NAD. Picks up hydrogens as well and becomes reduced NAD or NADH. Now, just when we talk about NAD, nicotinamide dinucleotide. Okay, each of these times there's 2 hydrogens being picked up. A hydrogen atom has a proton in the middle and an electron orbiting it. If you think back to junior chemistry, structure of an atom, you know atoms have protons, electrons, and neutrons. So hydrogen, atomic number of 1, has 1 proton and 1 electron. So if we have 2 hydrogens, there's our 2 hydrogens. Basically, when NAD picks up the picks up the 2 NAD is positively charged. When it picks up 2 hydrogens, so NAD, one of these has dissociated. So what do I mean by dissociated? I mean that the electron has moved away from the proton. So that's the H plus ion there, and the electron has moved away. This is just a bit of chemistry. You don't need to ever explain this. In fact, if you don't understand it, it's not a problem. But just in case you were wondering, what happens is one of these hydrogens here joins to the NAD. Sorry, that should have been D there. And so it becomes NADH. Now this was positively charged, so the electron in the other hydrogen also joins on, and the electron cancels out the positive charge. Okay, and so we get NADH, and we're left with this hydrogen ion over here. And so sometimes textbooks will say NADH plus H plus. That's where that comes from. Look, if that is all double Dutch to you, that's fine. Just forget about it. But just explaining why really there's 2 hydrogens being picked up here. If we wanted to be exact, that's what it would look like. Okay. So if we look at this, we want to tie things together. So far, what have we got? Well, if we look, we've started off here with glucose. then we've got pyruvate, then we've got acetyl coenzyme A, and we've ended up, where have those carbon molecules ended up? Well, actually, they've ended up there and there. Okay. What products have been made? So we know that the whole point of respiration is to produce ATP. So what about ATP? I'll highlight that in yellow. So up here, we had 2 ATP being used, but 4 made. That gives us a net profit of 2 molecules of ATP. In the link reaction, there's no ATP made at all. Forgot to say, in the Krebs cycle, there actually is 1 molecule of ATP being made. It doesn't really matter where I write it. I'm just going to write it there. So what do we have to remember about this? If we start off with 1 molecule of glucose, it splits into 2 pyruvates, and therefore we get 2 of everything in the link reaction and 2 Krebs cycles then going on. So what we're saying is, in terms of ATP, we would actually have 2 molecules, for every molecule of glucose, we're going to have 2 Krebs cycles. So we would have a grand total of 2 ATP being made there. Now, right at the beginning of respiration, when I was talking to you 2 weeks ago, we said there's 38 molecules of ATP. Right now, we're looking at 4. So there's obviously something else that's happening that we haven't thought of. So what have I not talked about? What's missing? Where's all the ATP being made? Sarah, you're going to tell me.
The electron transport chain.
Yep. So the electron transport chain, let me just come back to that. Where is the electron transport chain? Let me see. Camelia, where does the electron transport chain take place?
And the mitochondria. I don't know.
Yep, no, it is in the mitochondria.
Okay.
Anybody tell me exactly where it is in the mitochondria?
Matrix.
The matrix. No. Actually, it's not. The Krebs cycle is in the matrix, but the electron transport chain, where is it?
In the crystal.
Yep. So,
Miss, is it the
oh, right. Okay, so if we think of a mitochondrion, remember the inner membrane is folded. And basically, these molecules I'm talking about here are going to be put side by side on these inner membranes. Now, what I would do when I'm teaching this in school is take you out to the staircase outside, you know, the one where the drinks machine is, and I would give you all like stickers or cards. So one of you, for example, Ellen Hamilton, might put her near the top of the steps as with a sticker with flavoprotein on it. I might put Lydia next to her down a step on coenzyme Q, and Grace with cytochrome, and Zoe with cytochrome or reduced cytochromes, and maybe Hannah with cytochrome oxidase. Why do you think I would put you on the steps? What's the point there? What am I trying to get into your head? Any ideas?
Energy levels.
Yeah. So in your notes about the electron transport chain, it says that we've got these electron carriers. Now, sometimes they're called electron carriers, sometimes they're called hydrogen carriers, and we'll look at that in a moment. For now, don't worry too much. Basically, these molecules are carrier molecules, and they're passing hydrogens from one to the other. Flavoprotein is at a higher energy level than coenzyme Q, which is at a higher energy level than the cytochromes. So a phrase that we use to describe these electron carriers or hydrogen carriers is that they are at increasingly, or they're at progressively lower energy levels. So it's like going down steps, which is why I would get you out on the steps out there with your labels on or with your names on, and you'd arrange yourself in the right order, with flavoprotein being at the top and then coenzyme Q and so on. All right, so if you imagine that in your head, and basically what happens is another carrier molecule called NAD picks up the hydrogens and passes it to the flavoprotein. When the flavoprotein receives the hydrogen, it becomes reduced. So remember, reduction, a chemical is reduced when it accepts or receives hydrogens or electrons. Okay. What happens to the NAD if it gives up its hydrogen? Well, the opposite to reduction is oxidation, so it becomes oxidized. Flavoprotein passes the hydrogen on to the next carrier molecule, coenzyme Q. So the flavoprotein then has lost the hydrogen, so it's become oxidized. So we've got this series of reduction and then oxidation happening to each of these molecules in turn. And so that idea of reduction and oxidation, we call those pair of reactions redox reactions. And obviously you can see reduction, oxidation there. So the hydrogens are passed down. Now, at various steps in this, there's enough energy liberated, enough energy freed up to allow a molecule of ADP to join a molecule of phosphate and to make ATP. And that happens at 3 steps. Okay, it happens at 3 steps. So essentially, these electron carriers are fixed onto the cristae of the mitochondria, and NAD carries the hydrogen from glycolysis or the link reaction or the Krebs cycle, carries the hydrogen and passes it down a chain of electron carriers that are at progressively lower energy levels. At 3 steps, or at 3 stages in this, enough energy is liberated to join phosphate to ADP to make ATP. So for every NAD that enters electron transport chain, we get 3 ATP molecules made. Now, at this stage, where are we? If I highlight, I'll use green here. At this point here, the hydrogen splits. Now remember, we said hydrogen has the positive ion in the middle, and then it has an electron that's orbiting. So those 2 parts split up. And at this point here, it's the electrons that are passed down here. What happens is at that point, the hydrogen ion, it's just sort of floating around in here inside the mitochondria. But it will join up with the electron at the end. So if we've got an electron and a positive hydrogen ion, they join together to make up, they join together just to make up an ordinary hydrogen atom. Can anybody tell me what the final molecule that accepts the hydrogen is in this chain of events? What's the very last thing to pick up the hydrogen?
Oxygen.
Yeah, so that's really important. We need to have oxygen there to pick up the hydrogen, and obviously hydrogen and oxygen together make water. Don't worry too much about the numbers of hydrogens. I did sort of explain earlier that actually NAD really is associated with 2 hydrogens at the beginning. but it's sort of written as NADH plus H plus. Don't worry about the numbers. Basically, oxygen picks up the hydrogen and the final product is water. Whenever you learnt the word equation for respiration way back in junior school, glucose plus oxygen gives you carbon dioxide. Plus water plus energy. This is where the water is made here. This is the water. Carbon dioxide. Well, if we go back, hold on. There's carbon dioxide there, and there, and there. And the energy that we're talking about is ATP. Whoops. Okay. So we've talked about carbon dioxide, we've talked about water, and we've talked about ATP. So, all those times we talked about NADH being produced during glycolysis, link reaction, and Krebs cycle, those NAD molecules are carrying hydrogens and passing them to the flavoprotein or flavoprotein, it doesn't matter, you'll be writing it down anyway. Passes it, the flavoprotein passes hydrogen to the next coenzyme Q, which then basically the hydrogen splits, the electrons get passed down the rest of the carrier molecules. The hydrogens join up or associate again at the very last stage when the hydrogen is accepted by the oxygen. So the oxygen is the final hydrogen acceptor. Now, this is happening all the time because you're getting lots of NAD produced during the Krebs cycle. Add in, because the Krebs cycle, we had 3 molecules of ATP. Sorry, this happens 3 times with 3 NAD picking up hydrogen and making 3 NADHs. And remember, we've got 2 Krebs cycles going on at the same time. So we've got lots and lots of NADH that's being carrying the hydrogens to the electron transport chain. So this is happening all the time. The cell needs a constant supply of oxygen to allow this to happen. If the oxygen ran out, what's going to happen? Well, if the oxygen runs out, it means the next time NAD arrives at the top here, NADH arrives at the top there, it passes the hydrogens down here, 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 but there's no oxygen left. So this molecule here holds on to the hydrogen, it can't get rid of it. The next time the hydrogen's passed down, it gets this far, the next time it gets this far, and this far, and this far, and the whole thing grinds to a halt. So if there's no oxygen, basically the electron transport chain cannot function. So this doesn't happen during anaerobic conditions. Now, let me just see here. Right. One of the sheets that I gave you says describe how ATP is produced in the electron transport chain. That was one of the thing I put up this morning that had or yesterday that had the diagrams. I don't know when I put it up. I had the diagrams. It also had another page that had about 10 points on it. And I'm just going to read through that. If you haven't got it to your hand, that's fine. So it says hydrogen is carried to the electron transport chain by NADH and FADH2. So I've talked about NADH. You need to, as I say, know that this ATP production happens at 3 different stages. Okay? Happens at 3 different stages, as I've shown there. Now, so for every NADH that starts off the chain, we get 3 ATP. FADH2 arrives at a different place. So if I write FADH2, it cuts in after the flavoprotein. It cuts in there and passes the hydrogens on to coenzyme Q. So it's actually left out the first step. And that means that if it leaves out the first step, it means it's only producing 2 ATP, this one and this one. So NADH ends up with 3 ATP. If FADH2 carries the hydrogens, well, then we've only got 2 molecules of ATP being produced. Okay, that's really important. So to get back to the sentences, the points that I was reading out, we said hydrogen is carried to electron transport chain by NADH or FADH2. It's passed down a chain or a series of carrier molecules at progressively or successively lower energy levels. Now, I've really taken this from a mark scheme and adapted it a little bit to make sense. If you were asked in an essay, and look, we really don't know what's happening about exams, and I know it's unlikely that you have to answer questions in anything like this because they say that it's only the first 40%, but as yet they haven't told us. We reckon that, you know, we're going to have to set it. Well, certainly you'll do some kind of exam when you come back to school, whenever that is, but we'll give you notice about that. So we're just sort of going on ahead as normal. So if you were asked to summarize or to explain electron transport chain, and there were a lot of marks for it, this is the amount of detail they would expect. So hydrogen is carried to electron transport chain by NADH and FADH2. It's passed down a series of carriers at progressively lower energy levels, and you wouldn't be expected to have to name them all and all in the right order, as long as you say, for example, flavoprotein, coenzyme Q, cytochromes. Now, this is all very messy, but we said that at this point here, hold on, let me see. At this point here, I've sort of done in yellow, the hydrogen splits, or the hydrogen dissociates, and the electrons are passed down the chain. Okay? We've said that enough energy is liberated to produce ATP. The final electron acceptor is oxygen, and water is produced. And then what we said down here, if NADH enters the electron transport chain, you get 3 ATP. If it's FADH2, you get 2. And you could mention that at each stage, whenever the hydrogens or the electrons are passed down, the molecule that receives them becomes reduced and the molecule that gives them up becomes oxidized, and that's your redox reaction. Now, could you maybe write in the chat to let me know if you've any questions about that? Can I even see the chat? I don't know. Hold on here. Has anybody written in the chat?
No, I don't think so.
Right. Does anybody want to just shout if they want me to go over anything that I've said? Okay. That's Good. Either you all understand it or I've bored you and you've fallen asleep. Now, this diagram that I've given you has everything we've talked about. So, glycolysis. What I think would be good for you to do is to write 6 carbons, 3 carbons, 2 carbons, and then down here we've got 4 carbons, 6 carbons. Well, we haven't 5, it doesn't really matter. Carbon dioxide, carbon dioxide, another carbon dioxide. And the same on the other side. What I'm doing now is just highlighting any ATP. Now, the first one at the top is actually being used, so you need to be wary of that. And we've got 1 molecule in each of the Krebs cycles. NADH. I've used different colors for the NADH and the ATP. It's maybe not that clear on the screen. It's lovely on my iPad screen, but it's not so good on my laptop when I look at it. And finally, FADH2. Now, here's a table I produced earlier. What I want you to do is just get a scrap of paper and work out how many ATP, NADH, FADH2, how many molecules are produced at each stage, and then total them up at the bottom there. So if we start off with 1 molecule of glucose, just write in a number, and there's going to be zeros or dashes in some of the boxes, but put a number where it's relevant. Okay, so I'll give you a couple of minutes just to do that. Okay, I hope everybody's finished. So, let me see. I sort of want to miss anybody out, but it's hard when I only see some of you on the screen, and it's hard then to think of people that are not shown on my screen. I don't know how it picks you. Right, Hannah Sloss, how many ATP go in the first box top left? You're probably going to have to unmute.
I got 2.
Yep. Whoops. It'd be good if I had a pen. Grace Morrow's going to answer the next question. We're going to cross to the right. So yeah, there's 4 made, but there's 2 needed. So the overall is 2. So Grace, your NADH. How many NADH?
2.
2. And there's no FAD. Link reaction. Bethany, how many ATP?
I got none.
That's right. Very good. Ellen Hamilton, what about NADH? You're going to have to unmute.
2.
2, yeah, because there's 1 in each reaction, and remember there's 2 altogether. That's why I like that diagram, because it sets it out really easily to understand. Okay, there's no FADH2. Lydia, Krebs cycle.
2.
Yep, 1 per cycle. Grace Morrow, have I asked you? Maybe I have. I'll ask you again. So NADH.
6.
6, 3 per cycle gives us 6. And Zara, FADH2?
2.
Yeah. So if I can do mental maths, that's 4. I can do mental maths, I just can't write. 4, 10, and 2. So at the moment, we have got the grand total of 4 molecules of ATP. But yet I told you that there's 38 usually made. So where do we get the other ones from? Where is most of the ATP made? Just shout it out. Zoe, what do you think?
The electron transport chain.
Yeah. So for every NADH, we get 3 ATP. So that means we're going to get 30 ATP. For every FADH2, you get 2 molecules of ATP. So 2 2s are 4. And that gives us our total of 38 molecules of ATP. Now, that's the proper answer. You would actually say that, well, if we look at the next diagram, I think I've got the next diagram here. It's the one on the back, well, the second page. This is showing us glycolysis, link reaction, Krebs cycle. Glycolysis occurs in the cytoplasm. Oh, dear me. Okay, and then this all happens in the mitochondrion. And this happens in the matrix, or sorry, the cristae of the mitochondrion. So this is in the matrix of the mitochondrion here. And it actually needs 2 molecules of ATP, are quite often used to shuttle the electrons from the NADH. And so that's why in this diagram, the maximum is, if you use up 2 molecules, 2 away from 38, it's down to 36. Now, that's not something they really expect you to know. The electron transport chain, in that, it maybe hasn't come out very well in your copy, but it says electron transport chain. The ATP is made by oxidative, which means we need oxygen, phosphorylation. So adding on phosphate to something is phosphorylation. So we're going from ADP, adding on a phosphate molecule to make ATP, and we need oxygen for that to happen. Hence the term oxidative phosphorylation. The ATP that's made in glycolysis is called substrate level phosphorylation, whereas the one in the electron transport chain is oxidative phosphorylation. So we're getting most of our ATP made by oxidative phosphorylation at the electron transport chain. So hopefully that makes sense. Now, if we go back to this diagram here. So we've mentioned the electron transport chain, and that's really just everything that I talked about down there. We've got the flavoprotein, we've got coenzyme Q, and so on. Our final one is oxygen. They've drawn it in a slightly more complex way where the flavoprotein picks up the hydrogen, then becomes reduced, then it gives up the hydrogen to the next one. So the hydrogen molecules are sort of going down like this way here. Whoops. Doesn't matter, it's exactly the same as I showed you. And there's the ATP, ATP, ATP. And there's FAD coming in at this point here. Hydrogen's going, being passed down for this bit. Sorry, not hydrogens, the electrons get passed down for this bit here. The hydrogens sort of float about in the matrix and join in at the last stage. Now, we said that if there's no oxygen, this will not work. If you've no oxygen, it means that the NAD, if the NAD picks up hydrogen at any stage in glycolysis or the link reaction or Krebs cycle, it can't get rid of the hydrogen. And there's only a certain number of NAD and FAD molecules in each cell. They're not being made, they're not being made, they're just basically being recycled. It's like you have little people there and they pick up the hydrogens and carry them down to the electron transport chain. And the same here, and here, and here, and here. So if this electron transport chain can't work, it means we have massive problems, because it means that the link reaction can't work, the Krebs cycle can't work, because we're not having our NAD coming back for the next link, the next time that we need a link reaction to work. Does that make sense? Could you nod or shake your head so at least see whether some of you understand it or not? So it's as if, as I say, it was little people running around the cell. They're just carrying things around. They can't carry more hydrogens until they get rid of the hydrogen that they have already got. So if we have no oxygen, nothing below that line can work at all. Well, for a start, we need oxygen, for all of that as well. So none of that can happen, primarily because the electron transport chain can't happen. Essentially, we've got a massive problem here. I'm going to tell you glycolysis does work because there are other reactions, and obviously you've read about what happens in anaerobic respiration. But we're saying for glycolysis to continue, there needs to be some kind of mechanism to oxidize the NADH. So what happens if we look at this? At the end of glycolysis, we've got pyruvate. And we've got NADH. All right. What we want to do is to oxidize. This is reduced at the moment, so we want this to be oxidized here. So basically what we want to do is we want to move that hydrogen and add it onto the pyruvate. And that is actually what happens. Those two react and the NADH reduces the pyruvate to make ethanol. Molecule of carbon dioxide is made in the process, And if the NAD loses its hydrogen, it's oxidized. Okay, so basically the NADH loses the hydrogen, and we say the NAD is regenerated. Okay, so that's one way of looking at it. Another way of writing it. So the NAD has been regenerated. Pyruvate gets reduced and becomes ethanol, and a molecule of carbon dioxide is released. And so that actually, if you think back to GCSE, what organisms does this happen in? This type of anaerobic respiration. Zoe, can you remember?
I'm not sure.
Anybody? We use to make alcohol. Chantal, can you remember?
No.
Ellen Hamilton?
Yeast cells.
Ellen Black, that was very good. Yep, yeast. So this happens in yeast. It also happens in plants. So plants that are waterlogged, their roots will start to make alcohol actually, and that will eventually poison them. Which is why farmers don't want their plants to get waterlogged, and they dig drainage channels and put in drains and stuff like that. So glucose, way back in GCSE, you learned glucose in anaerobic conditions breaks down to give you ethanol or alcohol plus carbon dioxide plus energy. Okay. What are we saying happens? We're saying the glucose gets broken down. It goes through glycolysis. We get pyruvate. The pyruvate gets converted into the ethanol and a molecule of carbon dioxide. The NAD gets regenerated. And so the whole point of this happening is to allow glycolysis to keep on going in the cell. If that didn't happen, glycolysis would happen once, and then there'd be no more NAD. So to allow it to keep on going, this process here has to happen. So that happens in yeast cells or in plant cells. How much energy is there? Well, there's only 2 molecules of ATP. That's all. Because the NAD can't carry the hydrogens to electron transport chain because that's not working, and it won't go any further. It doesn't go to the link reaction. So there's only a very small amount of energy, but it's better than no energy. In your muscle cells, it's slightly different. Again, at the end of glycolysis, you've got pyruvate, you've got NADH. It passes its hydrogens onto the pyruvate and is regenerated or oxidized, regenerated, and you get lactate, which you might have called lactic acid. Lactic acid is sort of the old-fashioned name. It's what I called it when I was at school. Proper term is lactate. There's no carbon dioxide in this case. And yeah, same as before, just 2 molecules of ATP. So that is anaerobic respiration. So this is on this sheet that I gave you, and it just summarizes what we've said. Glucose to pyruvate. We've got our 2 NADH being made. Now, it's not a great diagram, but it was just in the middle of it. So because this is the same molecule. This is the NADH. It's come from there, and it's being regenerated, and so that then becomes that molecule there, and that can keep that will allow glycolysis to continue in the cell. pyruvate, ethanol, carbon dioxide, or lactate. And again, that's your NAD. So plant roots and yeast, exercise and muscle. Hopefully that makes sense. Any questions? Now, so this is a quick summary. We've got glycolysis here. We've got the link reaction, pyruvate to acetyl coenzyme A. Got the Krebs cycle at the bottom, and well, we would have the electron transport chain at the side there. But for now, we're thinking about how other substances, we've talked about glucose so far. Glucose is the main molecule that is broken down during respiration. You know from enzymes that you start off, the things that you start off in a reaction with are called the substrates. So this is the main respiratory substrate. Whoops. Okay. Now, you don't just survive on a diet of glucose. You will eat all sorts of things. So you've got carbohydrates, proteins, fats. Any of those can actually be used as a substrate for respiration. But in the most cases, it's glucose. If you eat a burger out of McDonald's, yeah, so in the burger BAP, you've got bread, so you've got starch there. Starch is a carbohydrate, so starch, any kind of carbohydrate, will get broken down, it'll get hydrolyzed into the monosaccharides. And so the monosaccharides are basically hexose sugars, and so they will enter, any carbohydrates will enter at this point here. If you eat proteins, so the protein in the beef in your burger, proteins are hydrolyzed into amino acids. Now, if you think back to the structure of an amino acid, we've got the amino group at one end. So hopefully that looks a little bit familiar. The R group, that could be CH2OH, it could be CH3, it could be, there's any sort of number of things. If we look at the other side and think about lipids, lipids will be hydrolyzed into glycerol. Glycerol looks like this, and these are all just hydrogens, C3H8. Sorry, C3, we've got, these are all hydrogens, the other side are HOs. Now, anytime we looked at this, I was keen that you would write things like 6 carbons, 3 carbons, 2 carbons, 6, 5, and 4. And that's because glycerol, as I said, it's got 3 carbons. It can join at this stage here. It joins just before the pyruvate stage. So if you've had lipid in your diet, so the fatty material in the burger, the lipid gets broken down into glycerol. Glycerol can slot into respiration at this point here. The fatty acids, are big long chain of big long hydrocarbon chain, lots of carbons there. We've got the acid group at one end. Sorry, that's not very neat. But all the rest, those are just carbons and hydrogens. So what happens to this molecule is it's hydrolyzed, it's chopped up into, I didn't do that too well, basically into 2 carbon units. So each of those 2 carbons, well, where is there anything with 2 carbons? Over here. They join at the acetyl coenzyme stage. and they will then go through this bit here. The glycerol goes there and, you know, goes around there. Amino acids, we said, well, they have, you know, they could have, that could just be, the R group might only have 1 carbon, in which case we've got a 3 carbon unit. So if that was the case, it would join at this point here. If it was bigger than that, well then if there were 2 carbons up there, that would be broken into, could be broken into 2 carbon units, so it could join at this point here. Or maybe it's bigger and we've got a 4 carbon block, it might join at that point there. Because amino acids are different, they can join it, they can be broken up and join it at a range of places in the whole process. Let's just go back to the fatty acids. If you had glycerol joining here and going around here, well, we're going to get how many molecules of, well, we're going to get 1 NAD here, 3 NAD H there, and an FAD H2, because remember it's only going around once. So that gives us 4 NADH and 1 FADH2. That gives us 2 ATP. Each of these gives us 3, so that's going to be 12. That's going to be 14 ATP. For the fatty acid, it joins at this stage here, and that's going to give us 3. Whoops. And 1 FADH2. So that's going to give us 9. That's going to give us 2. That's going to give us 11 ATP. But the fatty acid, that's going to happen again and again and again and again. A lot of fatty acids might have like 16 carbon molecules, which would mean that basically you're going around the Krebs cycle 8 times. Fatty acids yield an awful lot of ATP, and you can see why now, because each pair goes around the Krebs cycle and each pair is going to give you 11 molecules of ATP. So if you had 16 carbons there, break that into 8 pairs, and each pair gives you 11. That's 88 molecules of ATP. So that hopefully explains why fats can yield so much energy, much more than carbohydrates or proteins. Now, if we look back at this, there's another thing I want to bring out. We said if conditions are anaerobic, nothing happens underneath glycolysis. And if we look, that means that proteins cannot be used in anaerobic conditions, lipids cannot be used in anaerobic conditions. Okay, I know in some cases they arrive sort of at the pyruvate stage, but nothing happens to them. You know, there's no ATP produced after that. So basically we're saying if there's anaerobic respiration, it can only be anaerobic respiration with a carbohydrate as a respiratory substrate. So that's really what this diagram here is showing you. It's just a lot neater than what I ended up with, but it's the same idea. But I've explained hopefully how you can work it out logically, looking at the number of carbons that are there. Makes sense. Now I just try and get out of this and try and get back to normal here. Might take a wee while. Right. What's the best thing to do? Can you see me? Now, not on that stupid iPad thing. Doesn't really matter if you can see me or not. Doesn't, you know what I look like, so it's fine. Right. Maybe if you could turn your microphones on, and because I just want to ask you a few quick questions. So could you just either shout yes or no whether you actually understand? So do you feel you understand the electron transport chain okay now?
Yeah.
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 Yeah. Any aspect of the electron transport chain you'd like me to go through?
No.
No.
Okay. And anaerobic respiration, is that okay?
Yeah.
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 And using the different substrates?
Yeah.
Okay, look, what I want to do is go through the questions like you were doing. I'll maybe pick a, but I'm aware that we've been on for over an hour here. Have you got 10 minutes?
Yeah.
Mm-hmm.
Yeah.
Right. Well, then, I think maybe the question that people find most I'll pick it. Hold on. Maybe I'll share my screen again. Maybe if you turn your microphones off. Can you see my screen yet?
No.
No. Not so good. Hold on. Should be coming soon. Right. I think one of the questions that people find most difficult was that one there. Okay. So it says the diagram below illustrates molecular pathways within a yeast cell. Okay, so a couple of wee things here. Yeast, that's important. Small circles represent the carbon atoms. So what I would do is go, right, we've got a 6-carbon molecule there. That's pyruvate. So hence those are 3 molecules, 3 carbon molecules. And if we look, that goes through path Q to make a 2-carbon molecule. So Ellen Hamilton, any idea what the 2-carbon molecule might be? You have to unmute there.
I don't know. Is it
But if you look back at
I'm not too sure.
if you have a look back and see what happens to pyruvate normally,
It loses a carbon.
and what does it become?
A 2-carbon molecule.
Called
I'm not too sure. Acetyl-CoA.
Yep. So what we're saying is we've got pyruvate there. Yeah. Going through process Q. Sorry, I've just drawn over Q there. Giving us this 2-carbon molecule. So we're going to say that's going to be acetyl coenzyme A. Now, Ellen said that it loses a carbon. So this is it losing a carbon here. So that would make carbon dioxide. Yeah. If we go to the acetyl coenzyme molecule that I've just circled, it's going through this process here and it's making a 6-carbon and then a 5-carbon and then a 4-carbon molecule. And that 4-carbon says there's a pool of these 4-carbon molecules, and it's obviously joining with the 2. So hopefully you've worked out that this is the Krebs cycle down here. So if I pick a different color just to scribble on this, what we're saying is 6-carbon to make pyruvate. Well, that's probably glucose. To make pyruvate, well, that's going to be glycolysis happening down there. Q is the link reaction, and this is the Krebs cycle. First question asks you to name compounds X. So X is up here. This is X up the top there. So we've said, well, it's 6-carbon, it's probably glucose. And Y, so we haven't looked at Y. Y is this big long chain of carbon molecules which gets broken up into 2 carbon units. So hopefully you'll remember that, well, we talked about fatty acids getting broken into, fatty acids getting chopped up into 2 carbon units. So those would be fatty acids there for Y. Now, so this is a really difficult question. It came up quite a few years ago, but we like it because it makes you think sort of outside the box. It's a totally different kind of diagram to the one that you have in your notes, but it just has the same information. So we're saying this is glycolysis over here. This is the link reaction down here, and then we've got the Krebs cycle. The next question says P and Q are alternate pathways for pyruvate. So where's P? P, sorry, I've made this really messy here. So this is P over here, and then this is Q. So I've already said that Q is the link reaction. P is an alternate pathway. Well, what else can happen to pyruvate? Well, we talked about pyruvate being converted into ethanol or lactic acid or lactate if conditions are anaerobic. So it tells you this is a yeast cell. So hopefully you'll remember that in anaerobic conditions, pyruvate, anaerobic conditions, the pyruvate was converted into ethanol and carbon dioxide, the NADH, basically reduced the pyruvate to make ethanol. I'm stupid here. And the NAD was regenerated. So if we apply that knowledge, there's the pyruvate. It loses an oxygen, so that's another molecule of carbon dioxide there. So what must this be? This must be ethanol. So the question asks you, when will the yeast cell use pathway P? Well, that's whenever conditions are anaerobic. And what are the products of pathway P? Ethanol and carbon dioxide. Why is pathway P necessary? In other words, why does the pyruvate have to be reduced to make ethanol and carbon dioxide? Well, the whole reason for doing that isn't because the cell needs ethanol, or it doesn't need ethanol, it doesn't need carbon dioxide, but it needs to regenerate the NAD. Because if we don't regenerate the NAD, glycolysis won't happen. Now, my diagram is really, really messy, so I'm just going to move it up there. I'll move it across, get a wee bit of space here. Think back, glucose, pyruvate. What on earth happened there? Sorry. I haven't a clue what happened. Never mind. Pyruvate. To allow that reaction to happen, we have to have a supply of NAD to pick up hydrogens and become NADH. Normally, the NADH passes the hydrogens to the electron transport chain and comes back then in the oxidized form. But we've said the electron transport chain can't work because there's no oxygen. So we're just going to take that bit away. And we've got a problem because we've got NADH and we don't have any free NAD to allow this to keep on going down through glycolysis. So those two react together. The hydrogen joins onto the pyruvate to make alcohol. Molecule of carbon dioxide is produced and the NAD is regenerated as NAD. So we said, why do we need that pathway P to regenerate NAD to allow glycolysis to continue? Right. Again, I'll pick up that diagram of the question. I think I lost it there. Okay. The next question says, what is the eventual fate of the carbon atoms of pyruvate following pathway Q? So pathway Q, what is the eventual fate? Well, we said that that's where one of them ends up and that's where the other ends up. They're basically passed around there. That's in the Krebs cycle. So the eventual fate isn't the Krebs cycle. The eventual fate is they are released as carbon dioxide. Okay, I'll tidy the diagram up again. What is the molecule Z? Where is Z? Z is way over here. Well, if we look at that, it's Z is reacting with the pyruvate to make ethanol, we said. So what is Z? Z is NAD. The next question, where would you expect to find the pool of C4 acid molecules in a cell? Now, this was an older question, so they probably wouldn't give you the name oxoacetic. Let me see. Where would you find this pool here? Well, in other words, it's saying, where does this happen? So this is the Krebs cycle. The Krebs cycle happens in the mitochondrion, but you need to be more specific than that. So you need to say it's in the matrix of the mitochondrion. Finally, briefly explain how the combination of the C4 acid and an acetyl group eventually leads to the production of ATP. Now, there's 4 marks for this. We know that in the Krebs cycle, we get 1 molecule of ATP being produced by substrate level phosphorylation. So you will get 1 mark for saying that. What are the other 3 marks for? Well, and this is a very common type of question. You have to remember that in aerobic respiration, so where you've got the Krebs cycle, the link reaction and so on, most of the ATP is made in the electron transport chain. And that's really what most of your answer has to be about. So you get 1 mark for mentioning that 1 molecule of ATP is made directly in the Krebs cycle by substrate level phosphorylation. But that you've got NADH and H2 being produced, they carry hydrogens. So it's just that answer that I went through before, that they will carry hydrogens to electron transport chain. They'll be passed down a chain of carrier molecules at progressively lower energy levels. And you can name some of them. The oxygen is the final hydrogen acceptor and water is produced. enough energy is liberated at various stages to make ATP. If NADH enters electron transport chain, you get 3 molecules of ATP. If it's FADH2, you get 2 molecules of ATP. So that sort of series of statements that I gave you about the electron transport chain is what you want to be writing anytime it's asking you about, you know, what happens to NADH, where does it go in aerobic conditions, or where is most of the ATP generated, or explain how so much ATP is made. All right. Let me see. I think we will call it a day. Hold on, stop sharing. Ooh. I don't know, can you see me now? No, doesn't matter. Right, sure, you know what it'll look like anyway. I think like we've been on for an hour and a half and you're probably brain dead. I don't know. Are you happy to leave it there?
Yeah.
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 Mr. Chambers has actually put together a little video where he explains all of the I think he's gone through all of the questions. So I think I've just actually put that up while we've been on. I wasn't multitasking, I had scheduled it to come on sort of at the end. So like have a look at it and go through the questions, especially any that you've had difficulties with. If you've still got problems and you want me to go through something again with you, drop me a private message or send me a comment or something like that. All right? Okay. Hopefully you've remembered that you've got an application form to complete for the interview skills day. Does anybody have any questions about it, or anybody want to ask anything about it or talk or anything? No.
I don't think so.
I take it everybody is aware about it. Yeah. Look, and you know, it is really good practice for you. And actually, like, you know, every year people sort of coming out thinking, oh, I actually really enjoyed that. And it's going to be pretty different for you because of COVID and because you're going to be at home. But actually, some of you have already had online interviews and know what it's like. You know, so this kind of thing does happen in the real world because obviously people aren't coming in to do live interviews at the moment. So, like, strange if you were listening in, so we're just going to keep it one-to-one with the person interviewing you there. There will be a teacher sort of logged on in the background just for child protection issues, so don't need to worry about anything like that. All right, anybody have any problems with the form trying to fill it out?
No.
Okay, if you have, drop me a message. Right here, the next topic you are doing, I haven't said that much work because I thought like respiration is difficult. It's difficult to get your head around. The next one is photosynthesis, and it's actually pretty similar. Now Mr. Chambers' class has already started that. And so I'll be putting stuff up on Monday about photosynthesis. All right. All right, well, thanks very much for joining. Say hello to Karen.
Thanks, Miss.
Thank you.
Okay, right, so I will all let you log out and I will close up. All right?
Okay, thank you, Miss.
Thank
Thank
you.
you.
Bye.
Bye. Have a good weekend.
Oh, Camelia, are you still there?
Yeah.
In terms of your interview, what time is it right now?
9 o'clock.
9 o'clock at night. So look,
Yeah.
would you prefer to have an interview? If you were to have an interview next Friday afternoon, it would be like 10 o'clock at night. Would you prefer to have it the following week sometime in our morning?
Yeah.
Would that be better?
Yeah.
Because I reckon, I mean, you're probably a bit sleepy, are you?
No.
Because what I will try to do is maybe move a couple of the interviews till maybe Monday or Tuesday morning the following week, not next week, but the week after the skills day. So look, I will put up a few messages on. I'd set up a Google Classroom for all the Hong Kong boarders. So I'll maybe put a message up to see if people are happy to go with some other morning rather than the evening, because we don't want you all having to sit up late at night.
Yeah. Okay.
All right. Okay. Right.
Miss, can I use the form for you were saying?
Yeah.
Can I use you for the second
Yes,
referee?
definitely. Yeah, no, that's
I
fine.
know it's not, but yeah.
No, that's okay. No, put me down. There's no problem with that. That's fine.
Okay.
Okay.
Yeah.
How's-
Thank you.
How's Hong Kong?
Good. Just normal.
Are you allowed out?
Yeah.
Okay.
There are certain areas that they are not allowed to be going up to other places, but I'm safe.
Yeah. Are you allowed into other people's houses?
Yeah.
Right, we're not.
Okay. I know
Well-
you are in lockdown.
We're in lockdown.
Yeah.
You can sort of have a bubble, so you can go to one other house, but that's it. You know. So there we go.
Okay. Okay.
Any idea? Did you know that our schools are closed until the 5th of March?
5th of March?
Yeah, that was just announced last.
I haven't checked my email yet, so I'll check it now.
Well, it was only announced last night on the news, so yeah.
Oh, it was on the news, so okay.
Well, here, all the best. Hope you're keeping okay. All right?
Yeah.
Bye.
Okay. Thank you. Bye.